Have you ever stopped to think about how a single tiny cell knows what to do, or when to do it? Well, deep inside every living thing, there's this incredible world of microscopic machinery, constantly flipping switches on and off, reacting to the world. Today, we're going to take a look at those invisible switches, the tiny decision makers that are really running the whole show.
I mean, it really all boils down to this one question, right? How does a bacterium decide, hey, it's time to digest this new sugar, or for a pathogen, okay, now it's time to attack? And believe me, this isn't some random roll of the dice. No, it's a super precise decision made by this really elegant system of genetic controls.
Okay, so to get how one cell makes a choice, we first have to appreciate all the options it has to choose from. And for bacteria, that world of options is, surprisingly huge. It's actually shared across an entire species.
To really wrap your head around this, you have to split a species' total genetic info into two buckets. First, there's the core genome. Think of this as the essential toolkit, the genes that every single member of the species needs just to survive. But then, you've got the accessory genome. This is the cool stuff, the specialized genes for things like antibiotic resistance or weird metabolic tricks that only some of the individuals have.
So this diagram really puts it all together. That core genome is the part right in the middle, the common ground everyone shares. The accessory genes, those are all the unique add-ons that different members might carry around. And when you combine all of it, literally every possible gene found in any member of that species, you get what we call the pangenome. It's the total genetic potential of the entire population. It's massive.
Probably the best way to picture this is to think of a huge shared library. Every single bacterium has a copy of the essential reference books. That's the core genome. But they can also check out, borrow, and even trade these super specialized manuals from the accessories section. And that's what gives them brand new skills whenever they need them.
All right, let's zoom in on our single bacterium. Here it is, with its foundational circular chromosome. This little loop contains all those essential core genes it absolutely cannot live without. This is its baseline.
But of course, it doesn't just stop there. Bacteria are constantly grabbing new bits of DNA. A perfect example is something called a prophage. This is basically leftover DNA from a virus that infected one of its ancestors, and it gets stitched right into the bacterium's own genome. This new DNA becomes an accessory gene, a borrowed book from that big library. And boom, it instantly gives the cell a new power it didn't have before.
So we've seen the what, this incredible library of genes that's available. Now let's get into the how. How does a cell actually decide which books to read? We're going to look at one of the most classic genetic switches ever discovered. And this is really the whole ballgame, isn't it? Having 1,000 amazing books doesn't do you any good if you can't find the right one when you actually need it. The cell needs a system to turn genes on only when they're necessary. It's all about saving energy and responding perfectly to the environment.
Let's see how this works with the gene for digesting lactose, which is a type of sugar. Now, normally when there's no lactose around, this repressor protein acts like a big, heavy lock. It clamps right down onto a specific spot on the DNA called the operator. You can think of that as the keyhole. And by doing that, it physically blocks the cell's machinery from reading the gene. The switch is definitely off.
But what happens when lactose finally shows up to the party? Well, the cell cleverly converts a little bit of it into this molecule here, allolactose. And this little guy is the specific custom-made key that's designed to fit our protein lock.
So the key, the allolactose, finds the lock, that repressor protein. And when it binds, it causes the repressor to literally twist and change its shape. And that change forces it to let go of the DNA. It just pops right off.
And just like that, the roadblock is gone. The cell's reading machine, this thing called RNA polymerase, can now just zoom down the DNA track, making a working copy of the gene's instructions. The switch has been flipped on, and now the cell can make the proteins it needs to eat all that lactose. It's just a beautiful, simple, and incredibly effective system.
Okay, that protein switch is pretty smart, right? But what if a cell could just cut out the middleman completely? Well, it turns out nature has found an even more direct, even more elegant way to sense the world.
Just think about that for a second. What if you didn't need a separate lock and key at all? What if the environment itself could just reach in and directly flip the switch on the genetic instructions?
And this is exactly what an RNA thermometer does. Check it out. On the left, at a cooler temperature, the messenger RNA molecule, that working copy of the gene, is all folded up on itself, kind of like a zipper that's closed shut.
This folding hides the start signal that the cell's protein-making factories, the ribosomes, need to see to get going. But now, look at the right. As the temperature rises, the structure literally melts. The bonds holding it together break, the zipper opens up, and bam, that start signal is exposed. The factories can now lock on and start churning out protein.
It's just so direct and efficient. There's no middleman protein, no complex system. The temperature of the environment itself is the switch. You're watching physics do the work of biology right here. It's amazing.
And here's where it gets really serious. For a pathogenic bacterium, what does a sudden jump in temperature from, say, 30 to 37 degrees Celsius really mean? It means it just got inside a warm-blooded body. This temperature switch is the signal that screams, "You're inside a host. Time to turn on the attack genes and start the infection."
So, let's just quickly recap what we've seen. We went from the big picture all the way down to the tiny details. We started with the pangenome, that huge shared library of genes. Then we zoomed into the operon, a classic on-off switch made of protein. And finally, we saw the incredible RNA thermometer, where the genetic message itself changes shape to respond directly to the world around it.
From deciding what to have for dinner to knowing the exact moment to cause a disease, these tiny molecular switches are constantly making decisions that shape our entire biological world. It really makes you wonder, doesn't it? What other invisible, elegant mechanisms like these are out there right now, quietly running the show?